In this video, we are going to learn about lipids. Lipids, like carbohydrates, are also made of carbon, hydrogen, and oxygen. The main difference between lipids and carbohydrates, though, has to do with the amount of oxygen. If you remember back, carbohydrates tend to be multiples of this formula, CH2O. So 2 hydrogens for every oxygen. That means carbohydrates can have lots of oxygen. We do not see that with lipids. Lipids have very few oxygens. Mainly, they are carbon-hydrogen bonds. They're long hydrocarbon chains. And we know that carbon-hydrogen bonds are nonpolar bonds. So as a result, lipids are kind of grouped together because they are large hydrophobic molecules. So remember, hydrophobic means water-fearing. Nonpolar carbon-hydrogen bonds are not attracted to polar water molecules. Now, these large lipids are kind of grouped together into 3 main groups: fats, phospholipids, and steroids. We're going to go into each one in detail. The main thing to know about each of these molecules is that unlike the other macromolecules, these are not polymers. Carbohydrates are made of chains of monosaccharides. Proteins are made of chains of amino acids. Nucleic acids like DNA are made of chains of nucleotides. Lipids are not chains. They are grouped together because they are large hydrophobic molecules. Now, some of these lipids, such as fats and phospholipids, are made of smaller subunits, such as glycerol and fatty acids. However, they do not form chains. We do not form chains of fats in our bodies. We have independent fat molecules. We have independent steroid molecules. We have independent phospholipids. Okay, so let's learn more about the structure of lipids. So let's start off with fats. Fats are also called triacylglycerol or triglycerides. Now, they get their name because they have a glycerol, that is this gray portion here, which is a 3-carbon alcohol. They also have or 3 fatty acids. Now, a fatty acid is this yellow part here. You can see there are 3 of them. Now, fatty acids get their name because they are fatty. They have large amounts of carbons and hydrogens. So they're basically a large hydrocarbon chain, and then they have an acid. So you can see here in this dash part, that is the portion, that carboxyl group, COOH, that carboxyl group, that carboxylic acid group, okay, makes it an acid. And that is the portion that joins to the glycerol through dehydration synthesis to form this triglyceride. That is what we can see here. So you can see this yellow fatty acid, and here I'm going to circle the acid part, the carboxyl group. It is going to join to the glycerol through their hydroxyl group. So remember, whenever you do dehydration synthesis, you need to remove water. So you're going to need to remove a hydroxyl group, in this case from the carboxyl group on the fatty acid, and a hydrogen. When you remove those, you're able to then form the ester linkage, the covalent bond between the fatty acid and the glycerol. Because there are 3 fatty acids, that is 3 water molecules that are removed. Now, because of all those carbon-hydrogen bonds, fats tend to store lots of energy. That is their main job, is that they're energy storage. Now, we then have special tissue called adipose tissue where we can store extra fat for energy for future use. And that extra fat serves as cushions for our organs, and it also helps insulate our bodies. Now, let's just double check and make sure we understand about these large hydrocarbons. Okay, so here you can see we have 3 large hydrocarbons, those fatty acids. Okay, are they polar or nonpolar? They are nonpolar because of those carbon-hydrogen bonds. Although there are some oxygens which do form polar bonds, you can see in this giant fat, there are only 6 oxygens compared to many, many more hydrogens. So we tend to say this is a nonpolar molecule. And as a result, hydrophobic. Carbon-hydrogen bonds are nonpolar, making this molecule hydrophobic. So there are 2 main groups of fats that are found in nature. Those are saturated and unsaturated fats, and those have to do with the fatty acids. Now, when we look at this fatty acid right here, you guys see how many carbons and how each carbon is bound to 2 hydrogens, okay? Throughout, except for the end, which has the 3 hydrogens. Okay, so this fatty acid, we say, is saturated with hydrogens. It is full of hydrogens, okay? It's bound, every carbon is bound to the maximum number of hydrogen atoms. There are no double bonds between carbons in this. And as a result, your fatty acids are long and flat. So in this case, this is a saturated fat because this fat has 3 saturated fatty acids. Those fatty acids are flat. Because they're flat, they allow them to pack closer together. So let's think of each fat molecule as being a piece of paper. When you have a whole bunch of flat pieces of paper, you can make a nice solid stack. When you have a lot of nice flat saturated fat molecules, you make a nice solid fat. So when we think of solid fats, that would be things like butter and things like that. Now you want to limit these in your diet because they form solids, which can then stick to the lining of your blood vessels, and they can clog arteries, making it harder for blood to flow and oxygen to get to organs that you need to. Unsaturated fats, on the other hand, have double bonds. So if we were to say zoom in on part of this fatty acid chain, what you would notice is you'd have your carbons bound together, okay? And typically when a carbon is just bound to a carbon in a single bond, you can make 2 hydrogens. However, when it is double bonded to a carbon, it cannot bond to as many hydrogens, okay? Carbons make 4 bonds, 1, 2, 3, 4. When 2 of those bonds are locked into a double bond, that means less carbons are able to bind to hydrogen, or less bonds are able to form with hydrogen. So as a result, we say this is unsaturated. There are not a maximum number of hydrogens found. And so we can see that. Now, every time there is a double bond, it causes a kink. It causes a bend in that fatty acid chain. Okay? Now, because they're not flat, they don't pack as close together. So think about our stack of paper, each paper representing a fat. Well, if we crumple up those papers and we make them have lots of folds and kinks in that paper, like we see with unsaturated fatty acids, it's really hard to make a nice, neat stack of paper when they're all bent and crumpled. You can't pack them as close together. As a result, unsaturated fats tend to be liquid at room temperature because these fats cannot get close together to solidify. There's too much kind of space between all of those fatty acids. Now, unsaturated fats, it's referring to what is going on with those 3 fatty acids. An unsaturated fat could be monounsaturated. What that means is basically if you have your glycerol, you can still have 2 linear saturated fats, but then you have a unsaturated fatty acid. Okay, and so that creates a bump. Polyunsaturated is when you have multiple unsaturated fatty acids, like you see here. You can see how wild and crazy all those lipids are, all those fatty acids are, because they're all bent. That one's definitely not going to pack close together. Now, scientists figured out a way to turn unsaturated fats into solids, and those are called trans fats. Now, scientists knew that saturated fats, animal fats like butter or the actual fat on meat, is not good for you, okay? It can clog arteries. They knew that unsaturated fats, however, were much better for your health. So what they tried to do was they tried to turn healthy unsaturated vegetable oils, unsaturated fats, into solids. Think of vegetable shortening like Crisco. That is a vegetable oil that they have turned into a solid. Now they do this by messing with the hydrogens. Remember, unsaturated fats do not have the maximum number of hydrogens. By changing the location of hydrogens in these unsaturated fats and creating what we call a trans double bond, it caused these liquid vegetable fats to then be able to pack closer together and make solids. So here you can see in this picture, this is a trans double bond. You guys see how the hydrogens are located kind of on opposite sides of each other, whereas in this unsaturated fat, those hydrogens are on the same side, okay? And that kind of helps straighten out that fatty acid. We say that these fats are partially hydrogenated. So if you look on ingredients and you can see you're looking for a trans fat, it won't be listed as trans fat. It'll also often say a partially hydrogenated oil. Those are oils or unsaturated fats that they have messed with the hydrogens in order to get them to be a solid. Now, they did this because they wanted an alternative to animal fats and one that wasn't going to be as greasy. So think about oils. Oils make things greasy. Solid fats don't necessarily do that. And so they were trying to make a non-greasy alternative to form of vegetable fats. But scientists have since figured out that trans fats, like saturated fats, also tend to build up plaques and lead to heart disease. So you want to limit the number of trans fats in your diet. All right, now let's look at the next type of lipid, which is a phospholipid. A phospholipid is very similar to a triglyceride, except it does not have a third fatty acid. Instead, it has a phosphate group. So here you can see this is the glycerol, what I just put a box around. Okay, these yellow things down here, those are the fatty acids. In this case, one is saturated, one is unsaturated because it's got a bend. And we have our negative phosphate group, which we call the head. So oftentimes, when you see a cartoon version of a phospholipid, you'll see a head and two tails. Those tails are the fatty acids, and the head is referring to this negative phosphate group right here that's attached to that glycerol. Now, those negative charges on the phosphate group make this portion of the molecule attracted to water, while as the hydrocarbon tails are hydrophobic. And so we see that here when you add phospholipids to water. The tails aggregate together to try to hide from water, those hydrophobic tails. So if you put a bunch of phospholipids in water, they tend to form these bubbles called micelles. And you can see that there's kind of this dry interior where the hydrophobic tails are located, whereas the hydrophilic heads are very much attracted to those little water molecules that are floating around. Okay? Now, they also can form bilayers when in water. So again, we have dry tails on the interior, and when you have water on either side, you'll see the heads pointing to those. So this is what forms cell membranes. So the water outside the cell is referred to as the extracellular fluid, whereas the water inside the cell is referred to as the cytoplasm. We'll learn more about that when we learn about cells. So here you can see again, here's phospholipid bilayers for cell membranes. All right, our last group of lipids are steroids. They are not formed through the normal dehydration synthesis, like we saw with our phospholipids. So you can see the fatty acids are attached to that glycerol. Steroids are just really big hydrophobic molecules. They are formed with this fused 4-carbon rings, 3 hexagons and a pentagon. So here you can see, here's 1, 2, 3 hexagons, and then we have a pentagon attached. Now, different steroids are created by adding different functional group to this basic ring structure. Now, what you see here is cholesterol, okay? And from cholesterol, our body can build all kinds of different things, including sex hormones such as estrogen and testosterone. Now, cholesterol, we're going to learn, is going to be important as well in cell membranes. In addition to phospholipids, cell membranes contain another lipid, which is cholesterol. And we can see that here in this picture. Cholesterol has one hydroxyl group at the end. And so that part is polar, whereas the rest of this molecule is all carbon and hydrogen. So that's nonpolar. So you can see this little hydroxyl group is attracted to water like the heads of our phospholipids, whereas the bulk of the rings of the cholesterol are tucked in. So we'll see that when we look at cell membranes more in the next unit. In addition to cell membranes, our body needs cholesterol to turn it into other steroids that are used for communication, like sex hormones. However, like saturated and trans fats, they also form these plaques, like I was talking about earlier, these buildups of lipids in our blood vessels, which can lead to heart disease. Now, let's just look quick at sex hormones. We will talk more about these later as well. But here you can see that we have these hormones right here that are made in our adrenal cortex. So you have 2 kidneys located along your back wall. Your adrenal glands sit on top of your kidneys, okay? And their job is to make stress hormones like adrenaline, okay? So cortisol, aldosterone, they kind of work with your body to kind of maintain homeostasis when you're in kind of some of these kind of stressful situations or when things aren't necessarily in balance. And you can see they're very similar. They have that same cholesterol shape. They just have different functional groups added to them. Sex hormones are the same way. You have that same basic 4-ring cholesterol model. You just have different functional groups. So for example, testosterone, you have a carbonyl group. Estrogen, you have a hydroxyl group. And then progesterone, you see you have additional things and you have another carbonyl group located in a different place. Now, because these molecules have different structures, the body responds to them different ways, leading to different functions. We'll learn lots more about hormones and how they work later on this year, but it's important to note that some hormones are lipid-based, and those are our steroid hormones. So remember, lipids, large hydrophobic molecules. They are not polymers, but they all have large numbers of carbon-hydrogen bonds.